2073, the experiment of ultraviolet visible spectroscopy. My name is Chua Koh Seng, and I'll let my members introduce themselves.
Hi, my name is Fu Mei.
Hi, my name is Lo Wei Min.
And my name is Lu Yongming.
Ultraviolet and visible UV Vis absorption spectroscopy is the measurement of the attenuation of a beam of light after it passes through a sample or after the reflection of a sample surface. Absorption measurements can be a single wavelength or over an extended spectral range. Ultraviolet and visible light are energetic enough to promote outer electrons to higher energy levels, and UV Vis spectroscopy is usually applied to molecules or inorganic complex in the solution. The UV Vis spectra have a broad features that are of limited use for sample identification but are very useful for quantitative measurements. The concentration of an anything in solution can be determined by measuring the absorbance at some wavelength and applying the Beer's law. Now, let's move on to the operation process for the ultraviolet and visible spectrometer in our lab.
Let's show you a short video clip that shows you the operating process inside the casing of spectrometer.
A single wavelength beam will be emitted from the source here, and the other side is here. Then the beam will be passed through the reference cell and the sample cell. After that, both the reference beams and sample beam will be directed onto the detector.
There are usually two light sources for the UV visible spectrometer, one giving out visible light and one ultraviolet. This one is a tungsten lamp for the visible region, and this deuterium lamp gives out ultraviolet. This mirror directs the light from the appropriate source onto the monochromator. It contains diffraction grating that distracts the light, like the surface of a CD, and displays the light to its constituent wavelengths. Different wavelength corresponds to different color. Red is about 700 nanometer and blue around 400 nanometer. Wavelengths shorter than 350 nanometer are called ultraviolet. Shorter wavelength light has higher energy. The source produces white light that includes all wavelength and all color. The instrument scans through the spectrum, sending different wavelengths of light to the sample in sequence. This system is done by the grating, which rotates. A single wavelength passes into modulator, which mirror zones it. This chops the light into two beams, one beam passing through the reference cell and the other passing through the sample cell. So, this instrument is referred as a double beam instrument. Both reference and sample beam are directed by the mirror onto the detector. This compares the intensity and sends the signal proportional to the ratio intensity to the computer that controls the instrument. The logarithm of this ratio gives a quantity known as absorbance, which is a measure of how much light is being absorbed by the sample at that particular wavelength.
Now, I would like to elaborate on the application of UV Vis spectroscopy. First, qualitative analysis. UV Vis spectroscopy can categorize those types of compounds which absorb UV Vis radiation. Identification is done by comparing the absorption spectrum with spectra of known compounds. Second, quantitative analysis. UV Vis spectroscopy can be used for the quantitative determination of compounds that absorb UV Vis radiation. This determination is based on the Beer's law, which is A equals to negative log T and also equals to epsilon times V times C. Besides that, UV Vis spectroscopy can be used to detect the functional group of a compound and also the presence of impurities. Absence of a band at particular wavelength regarded as the evidence of absence of particular group. Other than that, UV Vis spectroscopy can be used as a detector of HPLC. Next, Kinetic of reaction can be studied using this technique as well. UV radiation is passes through the reaction cell and absorbance changes can be observed.
I'll try to summarize about the ultraviolet visible spectrometer. Spectrometer is useful for both qualitative and quantitative measurement, which there will be a beam of light passes through both sample and the standard solution, where it can be analyzed through the spectrometer. The application of the spectrometer included qualitative analysis, quantitative analysis, detection of functional group, detections of impurity, HPLC detector, criterion kinetic. That's all about our video. Thank you.